Deep inside every living cell is a high-density microscopic archive storing the exact instructions required to build and maintain an organism. This chart compares the varying scales of biological data by plotting specific genome sizes. The bacteriophage Phi-X174 operates on 5,386 nucleotides, while bacteriophage lambda uses 48,502 base pairs. Moving up to bacteria, E. coli carries 4.6 million base pairs. In these systems, the specific quantity of base pairs serves as a distinct molecular characteristic of the organism. To store these massive sequences of data, life universally relies on DNA. But early in evolutionary history, RNA existed as a highly versatile molecule capable of both storing genetics and catalyzing reactions. So why did DNA supersede it as the primary archive? This structural comparison reveals early life's vulnerability. RNA is built on a ribose sugar, possessing a reactive 2' hydroxyl group. This extra oxygen makes RNA easily degradable. DNA uses deoxyribose, lacking that oxygen, making it structurally inert. The nitrogenous bases present a second structural advantage. DNA utilizes thymine, which contains a stabilizing methyl group. RNA uses uracil, lacking that extra stability. Because of its chemical instability, RNA mutates at a rapid rate. Viruses carrying an RNA genome inherently have shorter lifespans and highly accelerated evolutionary cycles. By selecting for deoxyribose and thymine, DNA evolved as the secure, long-term vault for genetic data, freeing RNA to specialize dynamically as a messenger and cellular catalyst. The chemical stability of DNA solves the storage problem, but the sheer length of the polymer creates a physical constraint. A typical mammalian cell contains meters of DNA that must fit inside a nucleus measured in micrometers without tangling. Bacteria use a simpler solution, a region called the nucleoid, where negatively charged DNA loops are held together by positively charged non-histone proteins. Eukaryotes require a more complex architecture. This animation demonstrates the first level of eukaryotic compaction, the nucleosome. The negatively charged DNA strand is spooled by wrapping around a core of positively charged histone proteins. The histones acquire this necessary positive charge due to an abundance of basic amino acid residues that lock on to the acidic phosphate backbone of the DNA. Here we see a wider view of the resulting chromatin fiber, which does not spool uniformly. The regions that remain loosely packed and stain light are called euchromatin. The densely packed, dark staining regions are heterochromatin. Euchromatin is transcriptionally active, while heterochromatin is locked down and inactive. This physical packaging is the first mechanism cells use to regulate which genes are actually expressed. To utilize the active regions of euchromatin, the cell must copy the genetic information from the secure DNA archive into a mobile RNA format, a process called transcription. Both replication and transcription are energetically expensive. The cell relies on deoxyribonucleoside triphosphates, or DNTPs, which serve a dual purpose. They act as the raw substrates to build the sequence, and they provide the chemical energy to drive the polymerization. This schematic shows an unwound DNA transcription unit. The enzyme RNA polymerase dictates a rigid directional rule. It can only polymerize in one direction. Because of this rule, the RNA transcript is actively built in the 5' to 3' direction, using the 3' to 5' DNA strand as its physical guide. This guide is known as the template strand. This creates a nomenclature paradox with the displaced 5' to 3' DNA sequence, which is completely ignored by the polymerase. It is referred to as the coding strand. It earns that name because its sequence is perfectly identical to the newly synthesized RNA transcript, with the single exception of uracil taking the place of thymine. In bacteria, this RNA is ready for immediate use, but in eukaryotes, this primary transcript, heterogeneous nuclear RNA, or hnRNA, is non-functional. This diagram illustrates the eukaryotic split gene arrangement. Functional coding sequences, called exons, are physically interrupted by non-coding sequences called introns. The transcript is subjected to splicing, a processing step where the introns are precisely removed and the exons are joined together in sequential order. The molecule then undergoes capping, where an unusual nucleotide, methylguanosine triphosphate, is added to the 5' end. Finally, tailing occurs. 200 to 300 adenylate residues are sequentially attached to the 3' end in a template-independent manner. Only after splicing, capping, and tailing is the molecule classified as mature messenger RNA, fully authorized to transport the genetic code out of the nucleus. Outside the nucleus, the final step is translation, converting a sequence of nucleic acids into a sequence of amino acids to synthesize proteins. There is no natural complementarity between these two biological languages. Deciphering this process required treating the genome as a mathematical cipher. Physicist George Gamow calculated that a triplet code was a mathematical necessity to account for all 20 standard amino acids. This grid visualizes the parameters Gamow helped establish: 64 possible triplet codons mapping to specific amino acids. Within this matrix, 61 of the codons code directly for amino acids, while exactly 3 act strictly as punctuation to halt the synthesis process. These 3 are the universal terminator, or stop, codons: UAA, UAG, and UGA. The code also contains a crucial dual-function sequence, AUG. It codes for the amino acid methionine and simultaneously serves as the universal initiator codon, telling the ribosome exactly where to begin reading. The ribosome reads these triplets in a contiguous, unbroken sequence without punctuation. That absolute structural rigidity creates a severe vulnerability. A single microscopic error can derail the entire reading frame. Here is a side-by-side comparison illustrating a point mutation in the gene for the beta-globin chain. A change to a single base pair alters the triplet codon, replacing the amino acid glutamate with valine. This single substitution structurally collapses the resulting protein, causing the condition known as sickle cell anemia. The absolute fidelity of the genetic code is the mechanism that allows for complex life, but it also means a single nucleotide error can cascade into systemic, life-altering disease. Understanding how a single base pair causes disease required scaling our perspective out from individual triplet codes to mapping the entirety of the human genetic archive. Launched in 1990, the Human Genome Project was an international effort to sequence the estimated 3 billion chemical base pairs that make up human DNA. The researchers utilized 2 primary methodologies. The first approach focused on expressed sequence tags, or ESTs, identifying only the genes that are actively expressed as RNA. The second was sequence annotation. This was a blind, brute force sequencing of the entire genome, encompassing both the coding regions and the vast stretches of non-coding sequence, assigning function to specific regions later. Through this exhaustive mapping, the project arrived at a distinct conclusion regarding our genetic makeup. Any 2 human beings are 99.9% identical. To isolate the basis of individual variation, the search shifted from shared coding genes toward the non-coding sequences comprising the remaining 0.1%. These vast non-coding regions of the genome contain satellite DNA, repetitive units that do not code for proteins, but exhibit a remarkably high degree of polymorphism or sequence variation between individuals. This visualization shows many satellite DNA called variable number of tandem repeats, or VNTRs. A short sequence is arranged tandemly, but the repeat count varies drastically between individuals. Because of this, the physical size fluctuates heavily, ranging from 0.1 to 20 kilobases. By exploiting these highly specific size variations, scientist Alec Jeffreys developed the technique of DNA fingerprinting. This schematic outlines southern blot hybridization. First, sample DNA is digested into fragments by restriction endonucleases. Second, fragments are separated by electrophoresis and blotted onto a synthetic membrane. Finally, radio-labeled VNTR probes hybridize to complementary sequences, detected via autoradiography. The resulting autoradiogram displays multiple dark bands of varying sizes. Because the VNTR copy numbers are unique to the individual, the pattern of these bands provides a highly specific molecular barcode. From the chemical stability that protects the double helix, through the rigorous processing required to express its triplet code, down to the unique polymorphic stutters hidden in our satellite DNA, we have isolated and mapped the complete molecular basis of inheritance.